All right, welcome back. This is the second video for chapter 11, and again, we're talking about meiosis 2 this time. So this is the second half of that summary figure, meiosis 2, and in some cells, it's not shown here, but between meiosis 1 and meiosis 2, some cells go through a brief interphase called interkinesis. And this is a brief resting phase before they start the second part of meiosis. There is no S phase in interkinesis. There's no duplication of chromosomes. So after the two cells go through meiosis 1, they go through maybe interkinesis and then the series of events in meiosis 2, so that the chromosome number continues to really stay the same, except we're going to separate the sister chromatids by the end of meiosis 2. So what happens in prophase 2 really depends on what kind of cell or species you're looking at. If we're looking at human cells, it would look a lot like the picture that we see here, except instead of the 3 chromosomes we see here, we would have 23 in each of these cells. But if the chromosomes did decondense during telophase 1, they're going to recondense in prophase 2, and if the centrosomes were duplicated previously, then they're going to move to the opposite poles, so the opposite ends of the cell, and new spindles form, as you can see here. Similar to prometaphase 1, by the end of prometaphase 2, the nuclear envelopes will completely disappear again, the spindles will be fully formed, and again, each sister chromatid will have or create a kinetochore, and the microtubules will attach from the opposite poles. Remember, I drew this earlier. We have our sister chromatids, we have our centromere, and then the kinetochore proteins will be on the edges, will be attached to the spindles or the microtubules. Remember, microtubules are what make up those spindle fibers. Our book does a nice job showing how the spindle fibers, those microtubules, attach to our chromosomes differently in meiosis 1 compared to meiosis 2. So let's look at meiosis 1. This is prometaphase 1. Remember, in prometaphase 1, our homologous chromosomes are still attached at those chiasmata. And I can see the yellow centromeres. And I can see the spindle fibers attached to the kinetochores right there. In anaphase 1, the spindle fibers, when they shorten, will pull apart the homologous chromosomes. So homologous chromosomes are separated during anaphase 1, and you can see those chiasmata are pulled apart and the tetrads no longer exist because they're pulled apart. In meiosis 2, if I look at the difference between prometaphase 1 and prometaphase 2, you can see there are no more homologous chromosomes. It's just one chromosome here with its sister chromatid attached at the centromere. This time the spindle fibers are attached to each side of the sister chromatids. And in anaphase 2, we're going to see that the sister chromatids are pulled apart by the shortening of those spindle fibers. Metaphase 2 and anaphase 2 are really similar to mitosis, where the sister chromatids will line up at the metaphase plate. But remember, we're going to have 2 cells because we had 2 cells at the end of meiosis 1. Also in metaphase 2, remember, these are already haploid cells. So there are only one set of chromosomes. There's no homologue anymore. The homologue is in the other cell. So this is mom's hair color, for example, and dad's hair color would be all the way down here in the other cell. What we have is sister chromatids lining up in the equator, and in anaphase 2, the sister chromatids are split apart. So at the end of telophase 2, the chromosomes have arrived at the opposite poles. They're going to decondense, and the nuclear envelope will reform around the chromosomes. And in cytokinesis, the cells will be separated into a total of 4 unique haploid cells. Remember, the 4 cells are unique because of crossing over that happened in prophase 1 and because of independent assortment that happened in metaphase 1, allowing the cells to only carry either mom's version of a chromosome or dad's version of that chromosome. So let's summarize some of the key points that happen in meiosis. So in meiosis 1, in prophase 1, Remember, we had interphase happen already. So during interphase and S phase specifically, we created our sister chromatids when our chromosomes were duplicated. So I can see each of these chromosomes has a sister chromatid attached at the centromere. And in prophase 1, it's different from mitosis because we have synapsis. Remember, the two homologous chromosomes, or the pair of homologous chromosomes, come together, and they're bound together with that zipper-like structure, the synaptonemal complex. The chiasmata are where recombination or crossing over will occur, and crossing over occurs during prophase 1. In metaphase 1 is when we had independent assortment of the chromosomes, where the maternal and the paternal chromosomes could align randomly on either side of the metaphase plate, and each of these assorted independently from the other chromosomes. In anaphase, homologous chromosomes are pulled apart, and by telophase and cytokinesis, by the end of that stage, we have two unique haploid cells. So I should say diploid over here. Meiosis 2. So meiosis 2 is shown down here. This is a lot more similar to mitosis, but we start with haploid cells and we end with haploid cells. So the ploidy stays the same, kind of like mitosis. And something else that's similar is that we have sister chromatids that line up on the metaphase plate during metaphase 2, just like we do in mitosis metaphase. Sister chromatids are split apart in anaphase 2, just like we had in anaphase of mitosis. And this time we end up with 4 unique cells. Again, they're unique because of crossing over that happened in prophase 1 and independent assortment that happened in metaphase 1. This is the same slide I showed you earlier. Now that we've gone over the different phases, take a look at the outcomes and test yourself. See if you can cover up this part and talk about what happens in each of these stages of interphase and meiosis 1. And remember, by the end of meiosis 1, we already have reduced the chromosome number. We go from diploid to haploid cells. And I recommend doing the same thing for meiosis 2. Cover up this part of the figure and see if you can test yourself. Talk about each of these phases and what happens. And again, remember, in meiosis 2, we start with haploid cells and we end up with haploid cells. We end up with a total of 4 unique haploid daughter cells. And sorry, this is capitalized because of the font. I use some kind of title font here. It doesn't let me do lowercase. Although mitosis was on a previous exam, I do want you to remember the components so that we can compare and contrast meiosis and mitosis. So let's look at the two. Both of them divide the nucleus in eukaryotic cells. Remember, prokaryotes don't have a nucleus. They go through binary fission usually when they're dividing the cell. Mitosis, we're dividing only once. We get 2 cells. Meiosis divides twice, so we end up with 4 cells. In meiosis, we have homologous chromosomes pairing up, forming those tetrads that will cross over. That does not happen in mitosis. And in meiosis, crossing over happens between those non-sister chromatids of the homologous chromosomes. We also have random assortment of the chromosomes in metaphase 1, where the maternal and paternal chromosomes line up differently and independently of the other chromosomes, and those help to create genetic variation in the daughter cells. In contrast, mitosis produced genetically identical cells. Finally, meiosis reduces the ploidy of the cells. We start with diploid cells, 1. We end up with 4 haploid cells. And in mitosis, we don't change ploidy. So we have diploid to diploid, or in some organisms, we have, like plants, haploid to haploid. This is a nice figure from our book that compares meiosis and mitosis. In meiosis, I can see there are 2 divisions, right? There's meiosis 1, I get 2 cells at the end of that. And then by the end of meiosis 2, I have 4 cells. In contrast, in mitosis, I can see I have 1 division, ending up with 2 cells. I can see the chromosomes line up differently. In metaphase 1, they line up side by side. Remember, those homologous chromosomes line up side by side. And you can't really see it, it's too small, but there's crossing over during prophase 1. And I can see that meiosis 2 looks a lot more like mitosis. because in metaphase 2 of meiosis and metaphase of mitosis, those sister chromatids are lined up and they're going to be split up during anaphase. Our book also has a really nice chart under that figure I just showed you. This is very worthwhile to study for the next exam. So let's look at each process. DNA synthesis. It happens in S phase of interphase for both meiosis and mitosis. Synapsis of homologous chromosomes. Remember, that's when the homologous chromosomes line up really, really close together through that zipper-like complex, the synaptonemal complex. So it happens during prophase 1 of meiosis. It does not happen in mitosis. Crossing over, remember that happens when you form those chiasmata, and that happens during prophase 1 of meiosis, not in mitosis. Homologous chromosomes lining up side by side at the metaphase plate happens during metaphase 1, but does not happen during mitosis. Sister chromatids lining up happens during metaphase 2 of meiosis, and also during metaphase of mitosis, and that's why these two are more similar. So meiosis 2 is more similar to mitosis. Let's look at the outcome. So at the end of meiosis, we have 4 unique haploid cells, unique meaning genetically unique, and these are haploid again, even though we started with a diploid cell. In mitosis, we end up with 2 diploid cells at the end of mitosis, assuming we started with a diploid cell. In humans and other animals as well, genetic diversity is increased by a third component, random fertilization. So in humans, we talked about some of these before. There's genetic diversity because of crossing over that happens in prophase 1, independent assortment that happens in metaphase 1, and the new one that we're talking about now, random fertilization. What this means is that any sperm, and there are millions of sperm released, can fuse with any unfertilized egg, and these are called ovum. This is singular. Ova is plural. So the fusion of the 2 gametes, remember gametes are sperm and egg, each of them have about 8.4 million possible chromosome combinations. Remember we saw that earlier, 2 to the 23rd power because of an independent assortment. Each of these combinations can produce a zygote. A zygote is the first cell that's formed when a sperm and egg come together. So that gives us about 70 trillion diploid combinations. That is crazy. That's a ton. Crossing over adds even more variation to that random fertilization. So each zygote has a unique identity. And this is kind of something that you're going to study more in the second course of this series, 4B. But about 200 to 300 million sperm will enter the female reproductive tract when we're talking about humans. Sometimes people ask me about identical twins. So if you have someone or know someone with identical twins, what happened was one sperm fertilized the egg to create a zygote. And during development, one cell will rapidly go through mitosis. It'll form 2 cells, 4 cells, 8 cells, 16 cells. You'll eventually form a ball of cells. If those cells somehow separate during this process, you'll get identical twins. If you have fraternal twins, twins that are not genetically identical, usually what happened is you have 2 eggs that were fertilized by 2 different sperm. All right, and I wanted to show you this animation that summarizes the 3 ways we get genetic variation.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
All right, so that summarizes three different processes that increase genetic variation. We talked about independent assortment, crossing over, and random fertilization. That takes us to the end of our second video. In our third and final video, we're going to look at life cycles, and that is section 11.2 of the reading.